All right, so a couple of students asked me this morning where the appendix is in our book. Where are appendix in books? I didn't rat yourself out. You're ratting yourself out right now. In the back, in the back, in the back, right? So I had planned on going to the book today anyways, but let's do that. So how do we get to our book? So remember, everything is in modules, so we click on modules, and then we want to click on read, study, practice. No? Oh, I'm sorry. Thank you. I forgot to plug something in. My bad. That's why you didn't see your names either. Because usually you guys are like on the spot when I'm calling the names because they're up on the screen. Okay. We'll get there. And I had coffee and breakfast. Like, I have no excuse. Okay, so here's our book, right? And if you click here, it lists all the chapters, and of course down at the bottom is the appendices. Still not there. No, obviously not, because I didn't look over there. Help me out. Obviously, I need help today. Okay, so click here, right, where it lists all the chapters. Scroll all the way down to the bottom. You see appendices, right? So which one did I say to read? They don't have numbers. They're A, B, C, D, E, F, G. What are we learning about today? Go at microscope. So microscopy. B. And the reason I'm pulling up is because I'm going to refer to this image at some point in my lecture today. So, but we unfortunately, or fortunately, I don't know, it depends on how you look at it, need to finish the last bit of topic 1 before we can go on to topic 2, which was microscopy. I need to open both of these. So we finished up spontaneous generation. We had started on Kochs postulates. We only got through a couple of them, right? So let's start from the beginning for Kochs postulates. What section in the book should you read for Kochs postulates? Chapter 18, section 4. That's what 18.4 stands for. I'm not saying read the whole chapter. I'm saying read that one section of chapter 18. I did. Okay, good question. So where do you go for your PowerPoints and everything? Modules. Right, what module are we on? The first one. Number 1, right? Objectives in PowerPoints. So these are your study guides. So notice it says, this PowerPoint contains information not covered in your book, but you're still responsible for it, which means it's on the test. Right? For Kochs postulates, note chapter 1 refers to section 18.4 for more information. Read that section. Only that section in chapter 18, yes. Well, considering I haven't even posted it yet, not yet. Hence why I'm working this weekend, because this weekend I will make all the assignments for this module. So come next week, you can do them whenever you want, but you have to get them done before test 1, which is September 7th. Okay. This PowerPoint, too, contains information not covered in your book, but you're still responsible for it. Not that much that is not in your book. Very little. Read Appendix B, Toolbox 2.1, which is in Chapter 2. And in Section 2.4, read the parts Variation in the bacterial cell envelope. So that's why we're at it in our book. I'll just come back to this picture later. Read, study, practice, right? Chapter 2, Erica, 2. Really? 2. And you can even jump to sections, right? So I'd said section, what? 2.4. Right? The cell envelope. And specifically, I wanted you to read, when you got to that title, which I read this morning. Here's Toolbox 2.1. I don't know that you can skip to titles. No. I think I went past it. I was scrolling too fast. I should have known by what I was looking at. I need to read my own instructions again. The title, it just, see, there's a title. Plasma membrane. It just wasn't jumping out at me. Nutrient transfer. Bacterial cell wall. It's got to come up after that. There it is. It's right before the toolbox. The toolbox is embedded in it. It makes sense because they're related, right? So you would start here right above. That toolbox is a good place to find where you need to read. So you would read on till the end of that section, which isn't too bad. It looks like a lot on scrolling on the screen, but it's not a lot in your book. I don't have my book with me. Okay. I do have it in the trunk of my car. I was sitting and reading it before you know who showed up to do my nails. Because my Dwayne boy is watching your little one. All right, so any questions on that? Even I'm reading it, y'all, again, before I come talk to you guys. So if I'm doing it, I definitely expect you to do it. And until everybody nods their heads yes when I say, have you read the book, I'm going to keep torturing you. So read your book. I fought really hard to get a book that you guys can read. So read it. You will thank me come test time. Okay, so let's finish up Kochs postulates. So the first thing we need to do is all the organisms have to be sick with the same disease, right? Same symptoms, same signs. We need to get out that organism from all the people or all the organisms that are sick with that disease. We need to culture it in the lab and purify it so that we're studying a single organism, single species. We use a special technique called the streak plate method in order to do that, something those you guys in lab will do next week. Those you guys not in lab can watch my videos. Can we do this for all organisms? Can we grow them in artificial culture? No, right? We talked about Mycobacteria leprae that causes leprosy or Hansen's disease. They talk about that in your book as well, right? Where do they actually grow that in? to be able to study leprosy, since they can't grow it in a petri dish. What animal? Armadillos. Armadillos. They're on armadillos. Right? Then once you have it purified, you need to be able to give it to a healthy individual and it caused the same disease. Can we do this for all diseases? You've got to have a suitable host, right? If it's a human only, although HIV, human immunodeficiency virus, they actually can study it in monkeys, right? Because they're close enough related to us that they actually get human HIV. But what if it's something that we can't give to a mouse or some other animal like an armadillo and have that same disease? Can we satisfy this criteria? No, because no human is going to volunteer to purposely get a disease, right? I mean, people sign up for medical research. I think they're kind of cuckoo bananas sometimes for money, but you know what you're getting yourself into, right? There's a whole ethics issue, right? We can't just test it on people without their knowledge or consent or anything like that. It has apparently been done in the past, right? You hear these stories, it really did happen. Some early scientists who studied disease didn't become very popular because they would purposely give their friends things to see what happened. So, we don't do that. Which is why molecular science, DNA science, has really helped us with this, and why your book talks about the molecular postulates, right? Of doing this at a molecular level as opposed at the organismal level. So, and then we're not going to stop there. We want to be able to culture back out that original organism, and confirm that it caused that disease in the animal. So those are the 4 main steps of the criteria. He even knew when he was putting together these criteria that some of these conditions couldn't be met easily. But still, it's important. It helped us establish that link between the organism and the disease. And that's really important, because if we know if you have a particular disease, we can do what? Are we going to tell you're going to walk into the doctor's office and you have, I can't even think of an example. You have leprosy. Is the doctor going to be like, "Oh, you have leprosy, I'll see you next week"? No, hopefully what are they going to do? They're going to treat you, right? But if they don't know what you have, can they effectively treat you? No, right? So knowing what organism and therefore being able to treat our patients effectively is beneficial. And it all stems from the very beginning of knowing what is it that's causing what we see in our patients. And it's not always very easily detectable or obvious. And they a lot of times are making their best guess as to what's going on. There's a student in my genetics class that's friends with the high school student whose skin got burned off her body because they thought she had an infection or something, and they were giving her an antibiotic, and she had a severe allergic reaction to it. Sometimes we're not right. We don't have all the information. We make an educated guess, and we make a mistake. It happens. But of course, the more we study, the more we know, the more likely we are not to make those types of mistakes. That's why they say practicing medicine. It's not an exact science. It's not like, Star Trek, where we can put a scanner over your body and know exactly what's going on. We might get there someday. We're not there now. So, as I mentioned before this, in order for him to do this, the advent of agar had to come about. That special design of the petri dish to allow us to do artificial culture. The development of food, nutrient agars, and nutrient broths to feed the microorganisms. And methods for isolating them. That streak plate method, for one. There's a couple other methods that we'll talk about in the next module. So a lot of people ask, well, which guys do I need to know what they did? These are the guys, right? So we've already talked about Anthony. What is he attributed with? First person to see microorganisms, right? And he didn't even use like a microscope what we think of nowadays. His was a single lens. But he was so good at making lenses, he can get up to about 300 times magnified. That's crazy. Magnification for just a single lens. What about Reedy? What do we think about when we say Reedy? What did he help us with? Spontaneous generation. Did he help prove or disprove? Disprove. For what? Yeah, where the maggots came from was the flies, right? Not the meat turning into maggots. Pasteur, his list is kind of long, right? So his was obviously most people think of the swan neck flasks, right? But there's a couple other things we're going to talk about as related to him. And I always say to my students, if they truly ever invent a time machine, I want to go back and meet this one. I want to have a glass of wine with this guy. Because he was pretty freaking amazing, right? And of course, you know, pasteurization he came up with, and is why wine is so awesome nowadays, too. No, I don't even know if you could rank his contributions. They were so numerous and life-changing. Yeah. I mean, that's why I said there's a couple more we're going to talk about with him. So definitely helping disprove spontaneous generation, right, with the swan neck flasks and the other experiment he did with filtering the air through the cotton. We're going to mention in a moment a couple more. Tyndall and Cohen we already talked about, because even after Pasteur, people still didn't believe that spontaneous generation was false, right? These guys put the final nail in that coffin because they helped us know what type of organisms existed that were heat resistant, or what form of an organism. Endospores, right, which some bacteria can form, and you can't boil them to kill them. Koch, we just went over Koch's postulates. Lister, we're going to talk about in a second. What about Carl Woese? We already talked about him. What's his contributing factor that we learned about? What's his research led to? The 3 domains, right? And what are they? That one that starts with an A? Archaea. B? Bacteria. E? Eukarya. So Lister sounds like something you might have in your medicine cabinet at home, right? He didn't invent it, but it was named after him. Lister. Listerine. Listerine is what? Mouthwash, an antiseptic, right? Safe to use on your body, or in the case of your mouth. So in addition to the swan neck flasks and the help with spontaneous generation, Pasteur and others, like Rowe, discovered that the incubation of cultures for long intervals between transfers caused pathogens to lose their ability to cause disease. I call this the fat and lazy syndrome. Because when you grow them in artificial culture, do they have to fight for their food? No, you're giving it to them. They're like, here you go. In the real world, the reason why they harm us is because they're eating us, by the way. We are a big, fat, juicy cheeseburger to pathogenic bacteria. We are food. And them eating us can lead to the destruction of us, or at least parts of us. They're fighting against what when they're eating us? Our immune system. When they're in a petri dish, do they have to fight? No, again, we just give them food. Here you go. All by themselves. In their little home. No fighting. So they become lazy, right? They don't have to fight anymore. All the food's available to them. Right? They get fat like we would when we quit working. And so that ability to cause disease, they even start to lose because they're not using it. So my best example of this for people, how many people have seen Wall-E? I love that movie. It's so cute. The little robot in space. And all the people are on the spaceship, right? Because Earth has been like destroyed. And they all became fat and lazy, didn't they? And they were like riding around in their little rovers. And they had to start exercising, right? Because when gravity and having to walk around on Earth, they weren't able to do it at first. Because they didn't have to do it and didn't do it when they were in space. And so their bones deteriorated, they got fat, and they were having to recondition themselves to be able to adjust to life back on Earth. Our astronauts, of course, don't let themselves get fat and lazy, right? They exercise even in space. But long-term effects they've studied already in space, and there's no way you can replicate what happens on Earth to maintain muscle mass and bone mass because you don't have gravity. Artificial gravity even isn't enough. So that makes sense, you guys, right? So he used this hit to his advantage. So then they continued on, and they used this to make vaccines against chicken cholera, anthrax, even rabies. Being able to culture these organisms and making them less pathogenic. Not these big, ugly, mean bugs they used to be. They became fat and lazy. And so when we inject them into you, they've got to go back, and it's too much for them, right, to get in shape in that period of time. And your body, your immune system wipes them out before they can cause a problem, before they can remember how to be bad again, if it's even possible. They haven't even completely lost that ability. So when they say a virus, which I hate when they use that term, is live, it means it replicates inside of you, but they've usually modified that virus, so some of its pathogenic ability, its disease ability has been removed. So it can't cause the original disease it would. Bacteria a lot of times are weakened or killed or attenuated. Attenuated means they've been weakened in some way, right? Those really strong abilities that cause them to cause disease have been removed or modified. So they can't cause the actual disease in you. But gives your immune system the chance to say, hey, this is basically what the bad guy looks like. So when the bad guy comes along, the natural bad guy, you can defend against it. Because the awesome, amazing thing about your immunity is it has memory. It can remember certain things. So for some infections, you don't get them again because you're said to be what? Immune. That's why, because your immune system remembers it and attacks it immediately and eliminates it before it can cause a problem. You only get chickenpox once because once infected, you're always infected. The virus is just hiding and not replicating. It can reactivate, and that's called what? Shingles. It manifests in a different way because it's growing out of your sensory nerves where it was hiding. Instead of when you got chickenpox, you acquired it respiratorily, and it spread throughout your body. And that's why you get the skin rash. And again, typically you're only going to get that system-wide once because, again, your immune system's going to put it in check. But the problem with that is once infected, always infected. Little kids don't get it nowadays, though, because what do? We vaccinate. Can they get shingles? Yeah. So what's the shingles vaccine? It's a booster of the chickenpox vaccine. Just a lower dosage.
So if you have chickenpox, you're sure to get shingles?
You're not sure to get shingles. You could get shingles. Chickenpox reactivates when your immune system isn't keeping it in check. So numerous factors contribute to weakening of your immune system, right? One of the biggest factors, and so you'll see it even in So as we age, our immune system wanes. So usually you see it in elderly people. That's why they suggest basically getting a chickenpox booster as we age. But you'll see it in younger individuals like myself, 40, 50-year-olds. That's not a good sign to have your immune system at that age not be working properly. And it could be many different factors. A lot of times when several professors have gotten it here throughout the years, and those of us that are nerds walk around saying they have herpes, because it's actually the herpes class of virus. So, it's in that same class. Oh, it's very painful because it hides in your sensory nerves, right? The ones that help you feel. So then when it's growing and killing those cells and your immune system is attacking, it's attacking your sensory nerves. So it's extremely painful. Extremely painful. And they'll give you antivirals to help with it, but again, it's the immune system that's got to get everything back into check. So when people are extremely stressed or taking on too much, that's usually when you see it in younger individuals, and they're like, hey, you need to really reassess your life because you're doing too much and you're putting your body at risk. Yeah. Stressed. And then, they'll do additional testing because, again, if you're young and you're getting that's happening, again, there might be some immunological defect. We need to know what's going on because you're going to be more susceptible to other disease even.
So, just to be clear, I met someone and they didn't come out and straight up say that they had AIDS, but like, she had children.
Mm-hmm.
She was like, okay, I have an autoimmune deficiency syndrome, which basically stands for AIDS, right?
You know what? Can I table this one for now? Because we are going to get to it later. I promise you that. So that's a long discussion. It actually stands for acquired immunodeficiency syndrome. Because at one time they didn't know that HIV was causing your immune system to be deficient. So the term AIDS has become synonymous with HIV, but it's not. So I'll leave it at that for that right now. And I promise we're going to come back to it later. Okay. So Louis also demonstrated that alcohol fermentations and other fermentations were the result of microbial activity. And so why they didn't get the profiles they wanted with certain wines and stuff is because the wrong microbes were there. And they were causing the wine to turn into things that we didn't like. So he developed the process of pasteurization, mostly for the preservation of wine and during storage. So the other microbes, right? So after the fermentation happened, they would pasteurize it to kill all the microbes. So any of the ones that may have gotten in that would then turn it into something bad would kill them off or kill out most of them. So then it could be stored and not turn into something yucky. We use it for lots of other things nowadays, right? Especially dairy products. Juices, too, are pasteurized. That's why they can be on the shelf until you open it. Lister provided indirect evidence at the time that microbes were the cause of agents of disease. So he developed a system of surgery designed to prevent microbes from entering the wounds, as well as methods for treating instruments and surgical dressings. And because of that, his patients had, of course, fewer post-operative infections. So he recognized that this needed to be done as clean as possible. Nowadays, before surgery, like for my husband last week, they gave him special soap to wash his skin with, right, to decrease the microbial population that's normally present on your skin. So when they cut into his skin, they're not introducing those microbes that are naturally on your skin deeper into the body, right? They also, cleaned with iodine beforehand as well. They worked with sterile instruments that didn't contain any microbes to introduce them into the body. They put clean dressings on them afterwards. So that's it for